Hello everyone and welcome back to AP Bio with AP Playground. Today we're covering topic 5.3, Mendelian genetics. So as we first start talking about genetics, we'll be focusing on the work of Gregor Mendel, and we'll be talking about his laws of segregation and independent assortment, as well as the rules of probability that can be applied to genetics, and we'll be looking at Punnett squares and Mendelian patterns of inheritance. And so the suggested skills are statistical tests and data analysis, 5C, perform chi-square hypothesis testing, which we'll also be looking at, and argumentation, 6E, predict the causes or effects of a change in or disruption to one or more components in a biological system. So as we mentioned back in Unit 1, talking about macromolecules, and then in Unit 4 and 5, discussing mitosis and meiosis, genetic information is carried by DNA and RNA. These are our nucleic acids, and DNA codes for the production of proteins. And whatever proteins are produced lead to whatever outward traits an organism expresses, the appearance of the organism. We can also phrase this concept as the genotype or the genome affecting the phenotype or outward appearance. And the similarity in genome across the universe suggests common ancestry. For instance, humans share 98% of the same genome with pigs. So 98% of the DNA that we have, pigs also have, and there's only 2% difference that separates us from them. So this universality in the genome suggests that there's common ancestry, that we evolved from one organism. And when we talk about inheritance of these genotypes of these genomes, we're discussing heredity, which is the chance of offspring inheriting the traits of parents. So what are the chances that a certain trait from the parental generation is going to be inherited by the filial or the daughter generation? So I've mentioned the name Mendel a few times today, and now I'm going to explain who he is. So Gregor Mendel was an Austrian monk who did a lot of breeding of pea plants in his garden. And he made some discoveries about genetic inheritance through breeding these pea plants that he then documented, and that's how we have the modern study of heredity. So he determined that there were different versions of the same gene. And these were called alleles. And we've talked about these in 5.2. And one example of an allele is pea color. And there's some peas that are purple flowers, and some peas that have white flowers. And that means that their DNA is slightly different. They have a different allele of the same gene, which is pea color. And every sexually reproducing organism inherits one allele from each parent, which then contribute to make up the organism or the traits of that child. And the dominant allele is going to be expressed phenotypically, meaning that if, let's say, purple was the dominant allele, then when a purple organism that has purple flowers and an organism that has white flowers would be bred together, then all of the children would be purple. And when we express, and that's of course the phenotype, that's the outward appearance. And when we express the genotype, or how the genetic information is characterized, then the dominant allele is going to be expressed with a capital letter. And meanwhile, the allele that kind of steps behind, that isn't expressed outwardly, is going to be the recessive allele that has a lowercase letter. And they're different organisms. Some are homozygous, meaning they have two of the same alleles for a trait, so they're either both dominant alleles or both recessive alleles, or heterozygous, that have two different alleles for a trait, meaning one is dominant and the other is recessive. And this can be represented in a Punnett square. So we have both a phenotypic and genotypic representation of crossing the purple and white pea plants. So you can see here that when you breed a purple and a white pea plant together, what happens is you get 4 children that, or if you breed them 4 times, then you will get 4 children that all have that are all purple flowers. And I chose the letter C for color, and this is going to be our purple flower. It has our capital C's, and our white flower, it has our lowercase c's. And as you can see, both of these are homozygous parents. So this is going to have two dominant alleles, and the white flower is going to have two recessive alleles. And you can see that when they're crossed together within this Punnett square, which is how it's represented, you'll see that this child is going to get one of the dominant alleles and one of the recessive alleles. This one's going to get a dominant and a recessive. This one's getting a dominant allele and a recessive allele. This one's getting a dominant allele and a recessive allele. And as you can see, the result is that all four children are going to end up being heterozygous. But because the purple color is the dominant color, that is the color that will be expressed. And so this would be considered the parental generation, whereas this would be the F1 or filial 1 generation. These are the first children. And if we cross these purple flowers with one another, and they produce children or offspring or plants, then that would be the F2 generation. So Mendel came up with some laws to explain these observations. First one being the law of segregation, which says that if a parent has two of the same allele, then the offspring will all get that version of the gene. But if the parent has two different alleles, meaning they're heterozygous, genotypically, then each offspring has a 50% chance of getting one of the two alleles. So this just means that, let's say we have our homozygous dominant flower here, that during when we're doing this crossing, or when we're doing this breeding of two plants, then these alleles are segregated or separated. So then when we put them in our Punnett square, all of the offspring are going to have this dominant allele, because it'll be inherited by all of the offspring. But if it were to be a heterozygous plant, and we did the same thing with the segregation of alleles, then 50% would receive the recessive, while 50% would receive the dominant allele. So this law of segregation kind of gives a explanation behind the practices that we're using within our Punnett squares in order to determine the probability of each genetic outcome when breeding two organisms. And now, if you're not sure whether an organism is heterozygous or homozygous dominant, because of course both would phenotypically appear the same, but genotypically they'd be different, then you can use what's called a test cross. And a test cross just means that you're always going to cross that unknown organism with a homozygous recessive organism. So let's say in this case we'll call the gene A, so we'll cross it with 2 lowercase a, homozygous recessive, and if the organism were to be homozygous dominant, then all of the offspring would appear phenotypically of the dominant allele. That's the allele that would be expressed, because all offspring would be heterozygous, and so the dominant allele would be in control. Whereas if it was a heterozygous organism, then some of the offspring have the chance to express the recessive allele, because the organism will inherit 2 recessive alleles, and they will not all inherit a dominant allele, again, due to our law of segregation. And when you're crossing a single trait, that's just called a monohybrid cross. And if both, and generally what happens that when you cross a homozygous dominant and homozygous recessive organism, then you're going to, or not usually what happens, all the time what happens, is you're going to get heterozygous offspring. But if you're crossing 2 heterozygous offspring, such as here, then you're going to have a common ratio of 1 to 2 to 1 genotype, meaning that statistically you would have 25% that are dominant homozygous, 50% that are heterozygous, and 25% dominant recessive, talking about genotype, and then a 3 to 1 ratio of phenotype. So this 75% total would express the dominant allele outwardly, while this 25% would express the recessive allele. Now, there's a way to test whether the expected and then the observed outcome is the same, so whether or not there's a significant difference between these statistical expectations of 25, 50, 25, and then what actually is observed in reality, and to do that, we'll talk about it in a little bit, is called a chi-square test. But of course, we could be testing multiple characteristics at once, different traits, and finding the probability of that. And when you're doing two, now you have a dihybrid cross. And the phenotypic ratio there is going to be 9, 3, 3, 1, meaning that 9 are going to have the dominant allele for both of the traits. 3 are going to have dominant and recessive, and then the other 3 are going to have recessive and dominant, and then the last one is going to have recessive traits. And each one of these is going to have, or mostly each one of these is going to have a different genotype. So here we're crossing yellow and green peas. So that's the first trait we're looking at, yellow and green peas. But they're also smooth and wrinkly. So we start by crossing in the parental generation one homozygous dominant for both yellow and smooth, and then one homozygous recessive for both green and wrinkly, and that's going to get us the F1 generation, which is going to be entirely heterozygous for both traits. And then once these cross, you can see that each allele is going to separate, but at the same time, we're also going to have independent assortment. So each pair of alleles is going to segregate separately during gamete formation, meaning that the allele a cell receives for one gene does not influence the allele it receives for another gene. So that this means that having a smooth pea and having a yellow pea don't go together. Each allele is accounted for separately. It assorts independently. The gametes separate independently. So we have all these combinations. We have dominant, we have recessive dominant, we have dominant recessive, and then we have recessive recessive, because each one of these pairs of alleles is segregating separately during gamete formation. And now we're going to talk a little bit about probability rules. So there's two big words you need to know for probability. One of them is and the other one is or. And if you have a probability that's asking for and, so if you want the probability of this and this, then that means you have to multiply. Whereas if they're exclusively occurring events, so this or that, then you add the probabilities. So an example would be if you cross, these are our genotypes, if you cross AABB and AABB, meaning so we're crossing a dominant homozygous for AA and then a heterozygous for BB, and then two heterozygous both for AA and BB. What is the probability of getting a completely dominant heterozygous organism? So both homozygous dominant for A and homozygous dominant for B. So let's do the math. Because of our law of independent assortment, both of these we can find the probabilities of separately and then multiply, because each pair of alleles is going to segregate separately. So we can first cross our A's, meaning we're going to have capital, and then over here, capital lowercase. So that's going to create AA, and AA. So, and we're looking for two capital A's. We're looking for a homozygous dominant. So the chance of getting a homozygous dominant organism is going to be 2 out of 4, or 50%, or 1/2. Now let's calculate the probability for our B's. So we have our Punnett square, and we have our heterozygous B and our other heterozygous B. And so then we can do our Punnett square, or you might have realized by now that this is a heterozygous cross, meaning we're going to have a 1-to-1 ratio, and we're looking for a homozygous dominant, so 2 capital B's, meaning that that's only going to be 1 out of the 4, so 1/4, or 25% chance of happening. And now, because these are we're trying to find the probability of not one or the other, but both combined within one organism, we have to do a little bit of simple multiplication, and 1/2 times 1/4 gets you a probability of 1/8 for that organism that is going to have homozygous dominant for both A and homozygous dominant for B. So it's time to talk math. Chi-square test. Chi-square test helps you compare an expected to observed values in your data in order to determine if there's a significant difference between the two. So is the statistical probability that you calculated, does reality represent that, or is it significantly different from your calculations? And so your null hypothesis is always going to be that there is no difference, no significant difference between the two, between the observed and the expected. And if then the chi-square test shows that there is a difference, then you are rejecting the null hypothesis. They're saying there is a significant difference between the two. And so the formula for chi-square, and here's our Greek letter chi and the square, is the sum, or the sigma notation, meaning sum of all of the observed minus expected value squared over the expected value. So if you have, for instance, you're looking at 4 different data points, then you're going to go observed minus expected squared over expected, and that's your data point 1. Then you're going to add it to the same thing for data point 2, same thing for data point 3, and same thing for data point 4. That's just what the sum means. And so once you sum all values, you want to check your table at the 0.05 level of significance. That means that you have a 95% confidence in how, whether or not this is going to be a significant difference, and we don't really need to go more specifically than that for this course. So you're going to be using, and this is a diagram that will be provided on the AP exam, but you're going to be using this column right here that goes, that's 0.05 levels of significance. Then you also have to figure out your degrees of freedom, and that's going to be your value of options minus 1. So if there's like 4 potential things that could happen, then you're going to have 3 degrees of freedom. And then so you'd find at 0.5 in the degrees of freedom your critical value for chi-square. And if your value that you got from the chi-square test is larger than the critical value shown on the chart here on the table, then there is a significant difference between the observed and the expected data. Now let's put this into practice with an example. So let's do some practice. At a high school, students can choose to enter one of 3 doors. Custodians noticed that door number 3 was always getting broken and suggested that more students use that door because it has a hands-free opener. Science-minded students counted the number of students entering each door to see if the custodians were right. Door number 1 had 60 students enter, door number 2 had 66, and door number 3 had 80 students enter. Were the custodians right? So at first glance, it does look like more students entered through door 3, but let's check to see whether there's a statistically significant difference between the observed and expected value. So I already have our observed values for each door in here, and now let's think about what the expected value would be. So the expected value, if we have 3 doors that are all equally accessible, so there's an equal chance to go through each door. So if we have our same number of students here, which when you add it up, it becomes 206 students, statistically, if these are 3 equal options, then the equal number of students would go through each door. And if you divide 206 by 3, you get 68.67 students. So this would be our expected value, where an equal number of students go through each door. And yes, it does seem like more students are entering through door 3, 80 rather than 68.67. But using a chi-square test, we can see if the difference between the two is far enough. So next up, we have to do our observed minus expected values divided by expected value. So let's just do one problem. So our observed value is going to be 60. Our expected value is going to be 68.67. we square that, and the reason we square it is so that we avoid having a negative number, and then we divide that by the expected value. And I've already done this on my calculator, you can do so as well, but you're going to get a result of 1.0946. And once you do the same thing for the other options, you'll get for door 2 a 0.1038. And then lastly, we have for door 3 1.8694. And there you go. Now let's add up our number. So as a result, we're going to have 3.0678. And now let's take a look over here. What is our null hypothesis? Our null hypothesis is that there is no statistical difference. There is no door preference. And we want to see, are we going to accept this null hypothesis, or are we going to reject it and say the custodians are right? Door 3 is more popular. Now let's think about degrees of freedom. There are 3 options that students can take. They can enter door number 1, door number 2, or door number 3. So our degrees of freedom is going to be n minus 1. 3 minus 1 is equal to 2 degrees of freedom. There we go. Now we just need to take a look at our chi-square chart, and we're at already a level of significance of 0.05, and at 2 degrees of freedom, here's 1, here's 2, our critical value is 5.991. And let's compare our gotten value to 5.991. 3.0678, 5.991. 3.0678 is less than 5.991. Therefore, there is no door preference, there is no statistically significant difference, and the custodians are wrong. So the result is no. So there you have it. That's how you do chi-square. As long as you have your observed and expected values, all you have to do is plug them into the formula of observed minus expected squared divided by expected, and then add all of those values up and compare that value to the right place on the chart and see if it's larger. And if it is, then you've got yourself a statistically significant difference. So this was quite the video, and we've talked about Mendelian genetics and his 2 laws, segregation and independent assortment. In 5.4, we're going to talk about genetics that don't follow these laws. They are non-Mendelian. See you there.